Yeah. Okay, so I'm going to start anyway, and we may not need slides. My professor once shocked me because I was at the Royal Postgraduate Medical School at Hammersmith Hospital, sitting in the front row were all the professors, and in those days it was cutthroat. Your team won, so you as a resident had to come out and kill the rest. And if you didn't manage to kill them with your brilliant presentation, your professor would fight for you in the end. And the British trade unions decided to have a strike just at the moment as I started. So everyone, like that. And then the head of department, who everyone was trying to impress, of course, said, if you can't do it with a blackboard and a piece of chalk, you're not worthy of working here. And I simply said, but this is medical yes, sir. So I went to the blackboard and I gave my talk, and it worked very well. And I think we're about to start. So they're warming up. You see, I was right. My diagnosis was correct. Go away. Don't tell me. It's correct. So here we go. I'm going to talk to you about the medical informatics pillar. I don't think we should touch anything. We're there. Thank you very much. Leave it alone. Leave it alone. That's it. That's very good. That's there. Yeah, yes, that's fine. That's lovely. Thank you, gentlemen. Which shows that the directors of centres are worth something. Thank you. So I'm by profession a neurologist, and I've always done a little bit of clinical neurology throughout my life. But I've spent most of my professional life as a clinical scientist, essentially working at the mesoscopic level with imaging tools. But I'm a frustrated person, despite the outward appearance of bonhomie and energy and enthusiasm. I'm fundamentally frustrated inside. And the reason for this is that throughout my training at Cambridge, I could look down microscopes and see beautiful slices of anatomy of human brains and be completely seduced by them. It's why I'm in the profession. But there was no function. And my only access to function was a legacy from a great-great-grandfather of neurology whose name was Paul Broca and lived in Paris, and who had a patient who would say, "Dum." And when this patient died after 8 years in Sainte-Anne Hospital, in the night he did an autopsy, he took out the brain, he found a great hole here. And the following morning he presented the case at the French Society of Neurology and Neuropathology and suggested that the functions of language were situated in the dominant hemisphere in the frontal lobe. And so that brought function and structure together in neurology, and our life began. And we have continued. And we are now in 2013, and we were talking about 1860s, and I am now beginning to bang my head against the wall and not getting through. The pharmaceutical companies have been banging their head against the wall with us neuroscientists for about 40 years. They pulled out of stroke because we had the wrong models. We did it in the wrong species. We did it in lysencephalic brains. And we thought that this would have relevance to humans. They're now pulling out of dementia. And very soon they'll be pulling out of psychiatric disease, which is, of course, also a brain disease. So we really have a serious problem. I thought I might solve this because I was one of the first residents in the world to use an EMI CT scanner. But very soon it became clear that it still was only structure. And my information of function was second-rate. It wasn't second-rate, it was second-hand. It came from the patient who told me what he thought or she thought I wanted to know. And I wrote down what I thought that he or she was trying to tell me. And then I would put it in a drawer, and after 4 or 5 years I'd get out 6 or 7 of these, and I would write it up and send it to my friend who was the editor of Brain and say, "This is my syndrome, you just publish, please." And that I'd been doing for 150 years, and I was still doing it. So my only information about function was what the patient told me and how I interpreted what the patient told me. So then we had PET scanning, and that's when I got involved with imaging in a scientific manner. And there we had function at last. And this was mind-blowing, but extraordinarily difficult. And in the end, I dumped it. And I dumped it, why? Because MRI came along. And MRI came along, and it gave us structure initially, but within 4 or 5 years with the bold technique, it became clear that we could really, at a level of resolution that was important, get down to understanding what was happening at the mesoscopic level. So now we've used this technology for some years, 20 years or so, and I'm still banging my head against the wall. So I'm going to use the example of neurodegeneration. Why neurodegeneration? Because our societies are aging. At the age of 80, 20% of us have cognitive decline. We know that in Parkinson's disease, which is a neurodegenerative process, we need to lose 70% of nerve cells before we get our first symptoms or signs. So we're dealing with a redundantly organized organ. And we need to understand what's happening in Alzheimer's disease and the other dementias, as we've learned. You know, when I started, it was senility. Many of you remember cerebrovascular dementia, multi-infarct dementia, I'm sure, which has now disappeared completely. Then we went on to talk about dementia, then Alzheimer's disease. Then it became clear that there were other diseases that caused dementia other than Alzheimer's disease. Even trauma caused dementia. So we suddenly found a whole range of neurodegenerations, and we started to see the relationships between these and Parkinson's, Parkinsonian-like syndromes, because we looked at the proteins, and we found the same proteins present. And we started making inferences and got no much further. And why did we get not much further? Here we go. How do we try to prevent and treat Alzheimer's disease at the present? First question, can we diagnose it? Answer, no. We're wrong in 30 to 40% of cases, even in Tel Aviv University Hospital, even in the SHUV, even in Johns Hopkins, even in Harvard, even in the National Hospital for Nervous Diseases at Queen Square. We're wrong if we go to pathology at the end of life. And this has been confirmed this year with a couple of fantastic papers looking at all the pathology from the NIH Centers for Alzheimer's Disease that have been running for the last 10 years. Do the symptoms matter? Well, I've just told you that in Parkinson's disease you need to lose 70% of the cells before you get the first symptoms. So it's perfectly possible, and we now know pathologically that it is so, that many people between the ages of 60 and 80 have Alzheimer change in the hippocampi, in the medial temporal lobes. So people are compensating. So the immediate question then becomes, well, why aren't we treating those people? Well, we can't diagnose them. And why are the pharmaceutical companies making such heavy weather of finding drugs? Well, just imagine your target population, you've got a 30% error variance, and now suddenly we find that our normal control population has got an error variance we don't even understand, because there's Alzheimer change there, which is compensated. When I put those two error variances together and do a contrast, you need 3,000 in each arm. That's a lot of money. How many drugs are you going to screen in a year? So there are big problems. What weight do we give to pathology? At the moment it's the gold standard. In a moment I'll show you that perhaps it shouldn't be the gold standard. How do we compensate? What are the mechanisms for this redundancy? We've talked of plasticity. We've talked of plasticity at synaptic level, at systems level, at pathway levels, at biochemical levels, at neurotransmitter levels. We still live in a world where we use univariate statistics to do our studies of clinical efficacy. We look at correlations. We know nothing about causes. We're really in a big mess. What are the mechanisms which underlie this neurodegeneration? Is it the role of genes? Well, 10% of cases of dementia, some people say, may be autosomal dominant and have something to do with 2 or 3 genes that have been discovered. Are abnormal proteins involved? So amyloid, considered an abnormal protein until a paper in Science in 2011 that showed us that it's an antimicrobial in the brain. Are there abnormal foldings of proteins involved? We've known about prions for some time. We've heard the stories how it was completely impossible that prions could replicate, how this could be transmitted. Does that happen in Alzheimer's disease? There are papers beginning to appear, and I'll show you a little bit of evidence. Is it abnormal neurotransmission? You remember the first thing that people found was the loss of the cholinergic cells from the substantia nominata. The next thing was the amyloid. The next thing was the ApoE4. And each time the next thing has been found, we've piled in, we've looked for drugs. With acetylcholine, we have the most expensive placebos in the world. With amyloid, we've given people faster and more rapidly fatal diseases. And where we haven't, we've cleared the amyloid, but we haven't changed the cognitive profile. Where are we? We're still thinking in terms of Ehrlich's bullet. Single treatment for all these diseases, it's going to cure it? There's something very seriously wrong in our intellectual approach. Let me just show you this example here. This is the work of Kertész. This is 2005. This is the work that has gone on for about 10 years. He finds a group of dementias, frontotemporal dementia of the behavioral variant, very specific clinically. He follows 32 of these, and 6 of these end up with the same syndrome and multiple pathologies, of which one is normal. Now let's put in parentheses the gold standard of neuropathology. When a neuropathologist gets a brain, you think he examines the whole brain, don't you? Neuropathologists amongst you know that he goes and looks at the medial temporal lobe, examines that superficially for certain things, in greater detail if he has particular interest, and then says whether there is Alzheimer's disease present or not. So perhaps it's not such a great gold standard as we think. Now, of the remainder, 26 developed a second syndrome. Semantic dementia, progressive aphasia, corticobasal degeneration, a motor neuron disease. And of those, another 12 developed a third syndrome before they died. And again, the pathological changes were not particularly relevant. So we're in a mess. All right. Why do I think that this program is right for now? And why do I think it's feasible? Well, there are two reasons. It's right for now because we have the technologies. And it's feasible because in the last 10 years we have been witness to amazing changes in the enabling technologies. We're not even aware now of how amazing those changes have been. So let's start off with what we really want to do. I believe, and I put to you, that syndromic diagnosis has reached its limits. And what we want to do is to develop ideas about mechanisms and develop essentially diagnoses that are based on biological features and facts. Causal diagnoses. So this is a change in nosology. It's a development of something that's been going on for many years. There's nothing radical about this. As we add more and more diagnostic tests to our anamnesis, we're essentially doing this. We're trying to bring in more and more biological information on which we can anchor our diagnoses. So nothing radical, just speeding up the evolution. What's the first of these enabling technologies? That's the human genome. Now, many of us thought that we would get the human genome and that would solve many problems. These are, after all, the building blocks of organic matter. And as Henry has already shown you, a postdoc in half a day working in the National Library of Science in Washington using appropriate genetic and DSM-4 indexes describing all the known diseases of the brain can come up with a multidimensional map of this sort, which shows the various relationships between genes and different classes of disease. This is a very multidimensional map. It's here just as a figure. It's not worth, you can't even read it. But you saw a very similar thing in Henry's presentation. The problem is that now, 10 years later, after 10 years of GWAS, we're getting more and more papers saying, well, what has this produced? And as we think about it, we know that this was an extremely important development in science. But if we think about it, our variability is dependent on the same genes. There's a variation in those genes. Those genes produce proteins, transcriptomes, proteomics. Each of those how they develop are dependent on current context, current developmental state, current health state, nutrition, etc., etc., etc. So we're in a highly complex organization which we need to bring new techniques to bear on. So though this has been an incredibly important advance in our understanding of organic matter, it is insufficient in and of itself to give us the answer to our questions. Modern neuroimaging, which is my field at the mesoscopic level, imaging now, of course, spans all the way from single molecules up to behavioral imaging. This has become increasingly sophisticated and is giving us more and more information at the top end of the organizational pyramid, if you like. That end which is going to talk to us about consciousness, which is going to naturalize further and further philosophical concepts, and which is going to give us answers to some of the questions that our public wishes to know about. Our emotions, our mood changes, and the like. Now, one of the things that you must remember is this structure-function relationship and the way we can play with these using these modern MRI techniques. But something you may not be aware of, because those of you who are clinicians are used to using MRI in individuals to answer clinical questions, is that in science, thus far, pretty much, all we've been able to do is look at groups of subjects. And once we enter into groups, we're averaging, which is good for generating knowledge. But if we don't know how to create those groups, and I've just demonstrated to you that very often we're very wrong when we classify people clinically into different groups, then this becomes much less of an important, or at least a trenchant, way of looking at things. Here you see two types of diseases of the degenerative type, Alzheimer's on the one side, a semantic dementia on the other. You see the different patterns of atrophy, the classical medial temporal patterns, hippocampi, posterior temporal parietal regions, and so on, which have now been demonstrated to be classical for groups of people with Alzheimer's disease. And here is the semantic dementia with atrophy focused on the dominant temporal pole, but also affecting the mesial temporal lobes on both sides. So you can see where the confusion can come in, because there's overlap, but you can also see where the individual variability can come in. These sorts of patterns have become more effective at showing up different types of dementias that affect the same clinical function. So, for example, language. There are now different types of dementias being demonstrated, giving different types of language disability. Some attempts at doing studies in individuals have been helpful, but actually the statistical basis for these is poor, and therefore they have not become universal, but occasional extremely interesting facts have emerged. For example, this lady who started off with her dementia and some atrophy in the dominant temporal lobe, temporal coming into the insula, who over the course of yearly scans shows a progression of this atrophy which is spatially concurrent from the site of origin and affects basal areas which are linked to these cortical atrophic areas. So perhaps here is a suggestion that there is something that is spreading. Perhaps here is information that suggests we should be looking at transmissibility and should be looking at prions or prion-like particles, at the proteins in different ways. With PET, we've been able to show that amyloid is present in large amounts in minimally affected people before the dementia sets in. So either there's some very strange way in which this large load of amyloid causes the Alzheimer's disease, or it's a tombstone. It's a tombstone, and when it's killed enough of it, when enough of the nerve cells have atrophied, have died, have degenerated, have been stuffed full of amyloid, we begin then to decompense and become demented. There have been ways of trying to put together the different types of imaging into multivariate analyses. There's some beautiful studies coming from San Francisco from Bill Seeley, where he looks at structural anatomy, finds areas of atrophy that typify the earliest stages of particular syndromes, uses steady-state functional imaging to look at how these atrophic areas interact with other areas in the steady state and how that differs from normals. Look at functional connections by looking at correlations of local concentrations of grey matter in the brain from using as seed regions the primarily atrophic regions, and then trying to put this all together into single patterns. And as you can see, you can find patterns in groups of patients again which match the different syndromes. So certainly there's something in this in terms of a specificity for the syndrome. I'm not saying that syndromic diagnosis is all wrong. I'm just saying that it doesn't give us sufficient information in order to take the topic further. One piece of knowledge that this provides, however, is that the dementias, the different types of dementia, appear also to be diseases of brain systems. And therefore the systemic approach at this mesoscopic level may also prove fruitful in the end. At the moment, the advances in imaging are being made in two areas. The first is the imaging is becoming more quantitative and multiparametric, so that in a single 18 minutes one can take images of different types which give you quantitative aspects and information about the quality of the tissue, its content of iron, its content of myelin, the transmission of fibers through areas. We don't know much about the numbers of fibers yet, because we haven't got analytical techniques for that. But we know about water content. We know about water mobility in absolute terms, so that we can begin to put together databases where studies from different machines, from different centers, can be brought together. This is extremely important. The application of these sorts of techniques have allowed us to demonstrate endophenotypes, and they've allowed us also to prove this notion of redundancy. So that using the degenerative dementia known as Huntington's disease, where you can make an independent diagnosis with a genetic test which gives you 100% prediction of whether the disease is present or not, we've taken scans from our colleagues in Tasmania, where there's a lot of Huntington's, scans of normal people in Huntington families. And we've divided these single, simple clinical scans up into two on the basis of whether they do or do not have the appropriate mutation of the Huntington gene. And when contrasting these scans from normal people, we find that there is already here in yellow atrophy, which is distributed specifically in regions of the brain that we know affected by this neuropathology. In the caudate nuclei, in the tails of the caudate, in the tectum of the brainstem, places which are responsible for the initial changes, the movement disorders, the changes in rapidity of movements of the eyes in a saccadic fashion in the vertical direction, and so on. So this atrophy is present in normal people. That is a fact. Secondly, we can show that with the same phenotype, but with different genetic background, there are differences in anatomical configurations in the brain. So one phenotype is called dystonia. These are these extraordinary changes in muscle tone which provide these postural differences, sometimes in the face, the hands. Musicians can sometimes develop them, and so on and so on. Many of them are under genetic control, and under genetic control through mutations of a dominant type, but which are only partially penetrant. So you can have the mutation with and without the dystonia, and you can have the dystonia with another mutation, or you can have nothing at all with the other mutation. So you have a four-square design, and you can look at the interactions between the gene and the phenotype. And that's what you see here, a clear interaction between the normal gene, the DIT1 gene mutation, which results in the progressive atrophy of the basal ganglion of the putamen in this common type of dystonia. But in another dystonia, due to another gene, there's a progressive hypertrophy of the putamen. So using these techniques, we can delve into the endophenotypes, get a bit closer to the biology, but again, we're dealing with group studies. So group studies require samples which can be faulty, averaging, contrasting, often use univariate statistics, though as soon as you get into interactions you can look at nonlinear changes, allow generalization and provide us with knowledge. What we really want to know is what's happening in the medical side for individuals, because the drive is towards personalized medicine. The drive after knowing the genome is to help individuals and help them recover from their diseases. And here we've been using modern mathematics, modern statistics. We've been using the second major change that's occurred in imaging, which is that of progression on the analytical side. And on the analytical side, we've been moving to the use of things such as machine learning techniques. And in particular, I'm going to show you some results from support vector machine classifications of these individual images of brain structure that I've showed you. And these classifications may be binary, they may be probabilistic, they're potentially multivariate, and they should help with diagnosis and or prognosis, so that in the long term what we would wish to have are groups of 20 normals and 20 dements of the same type, biologically defined, so that every person that comes into a university clinic and deposits their data on the university databases can act as a subject not only for research into these diseases but also for screening for drugs. If you only need 20/20 in order to say there's a significant effect or not, you're way on the road to bringing back the pharmaceutical teams. So here are the results of a study that we performed on patients with Alzheimer's disease and age-matched normals. So you remember the problem. So in order to overcome that problem and try and get a gold standard set of data, we asked friends around the world to send us scans of their patients who had died and had had post-mortem and were shown to have Alzheimer's change or not. And then we began contrasting them. And we also got the classical scans from clinically diagnosed Alzheimer's and normals. And the result here is 80% correct, terrible sensitivity, not terribly good specificity. That's what we would expect from the studies I talked to you about at the beginning, the pathological studies. We've done this with two groups of such scans, gold standard scans, and you can see the results. Massive change in specificity, sensitivity, correct classification, all with one scan. Think of the cost implications if this were to become something that was adopted as a diagnostic technique. Instead of coming into hospital for a week and seeing all the specialists, you could have one scan and you would know whether you dropped into one or other classification. And we've developed this a little further. We've shown formally that it's better than the eye of the radiologist, who finds it difficult to even conceive of the multidimensional problem that you have here. This is not a single lesion. This is a big multidimensional problem. We've even predicted scores on psychological tests, measured them in people, and seen what the correlations have been on the image predicted scores of tests and on the actual scores, and shown that some perform extremely well as predictors of disease, some are lousy. Let's kick them out for starters from our screening tests. And then in a group like this, you can see there are some who do better than their prediction, there's some who do worse than their prediction. Surely these are the people we should be investigating in order to discover what is the biological basis for a better degree of compensation or an inability to compensate. We're now getting into the basal ganglia architecture with high specificity, beginning to show in relation to cognitive features situations in the anatomy where the separation, the functional specialization of areas and their fibers, which are kept separate in the anatomy of the brain, one of the great mysteries of the human brain organization, which we don't understand terribly well, how these primary features, sensory features and motor outputs that come in and go out, which are kept separate along long stretches of pathway, where we get the integration. Well, in the basal ganglia we find areas where there's now overlap. And we find not only long loops, as described in the past by Long and his colleagues, we also find short loops. So we're beginning to find architectures which need to be brought into this organization. And then just to bring you back to data mining, back to the sort of thing that I previously showed in relation to the genomics. Here is an area of the brain of intense interest at the moment, the subthalamic nucleus. because people are putting needles into this area and pacemakers in order to stop Parkinsonian tremor, in order to treat Parkinson's disease. Whereas in these situations, what we're finding is that the literature tells us that there are many, many more connections than the ones that we had suspected, simply in terms of the models, the simple models that attempt to describe why this therapy is useful. Our connections in blue, efferent, and in red, afferent, these connections have all been now replicated using predictive tractography on the basis of DTI. So this technology is going to build bridges between the genomic and the cognitive. What about modern neurosciences? Why haven't they helped us more? This has been largely treated by Henry. My rather criticized explanation is that the fact that we've been so good with reductionist science at concentrating on individual methods that we've developed. These are all methods in development. It's a small sample. And the depth in which we've gone have meant that we've worked in a completely unholistic space, a space which is fragmented, as to use Henry's word. We don't have a framework. You could call the framework a theory if you like. You can call the theory a model if you like. But somehow, somewhere, we need to know how the different bits of knowledge link to each other. This is why the Human Brain Project exists. And I'm now just going to briefly explain to you how these various aspects of medical development and medical evolution are going to interact with that particular project. One of the most important of these technologies, however, is modern informatics technology. And this is truly explosive in development. 10 years ago, we could not have thought of the Human Brain Project. Just look at the various problems. We've talked about some of the problems of computing. Nevertheless, we can construct exabyte computers. So the necessity for the sort of computing that is required for the Human Brain Project can be delivered at cost. The big cost being the energy. So the energy limitation beyond exascale is not very well recognized, and that's one reason why we have the third division, which isn't represented today, which is that of computer science, which is absolutely critical both to the first and second divisions, the neuroscience and the medical neuroscience, but also is interested in its own right in the human brain as a way of trying to inspire new architectures, new technologies in computing. But all of you use the internet. Who heard of HTML5 10 years ago? Oracle. In the last 10 years, it's lost the America's Cup to the Swiss Navy twice. No idea that it could be so powerful right now. It's just got it back, you remember. The cloud environment. Who was using Dropbox 3 years ago? No, one person. Very good. You're obviously an adventurer in this field. Database querying and addressing. We all do it all the time. He's doing it over there on his Google. You know, you just put in your parameters. You might have a Facebook page, and you've got access to everyone's data who's plugged into the network. So we need an infrastructure amongst hospitals in research databases, which will allow us to create a similar sort of network where people who want to answer questions will be able to go out and find results in the existing data throughout all the hospitals of the world, which at the moment are totally unused. They're there for legal purposes, medical legal purposes. When I go to the Minister of Health in Switzerland and I say to him, for the Swiss franc that I pay you to do me a brain scan and put it in a PACS machine, you could also have a data set for developing Novartis's new drugs. They're onto it, dead quick. Politicians understand that. The Swiss are transferring patient data anonymized to Novartis now. So all these issues of privacy and so on are solved, or if not actually solved, solvable in the context of this sort of work. So it is extremely important that we understand that we are doing it now because it is now possible. So we've now got to a stage where we have modern information technology, But the other thing that we have is a facilitated calculation power, which has bred a whole new area of mathematics, complexity mathematics, which is maturing extremely rapidly because there's feedback from the calculations and validations of what's happening. So we're in a position now to start asking questions about mechanistic and causative diagnosis. The high-level challenge is a paradigm shift. Can we do it? Can we move from phenomena to biology in defining disease? And this is our roadmap. Our roadmap in medical informatics is to start with federating data, and that's our first 2.5 years. So this involves putting in a server, possibly a copy data store, so that there's no movement and no corruption of the data on the hospital databases or in your research databases. You're given a contract, you join in, you get access to everyone else. You don't join in, you play around with your own data, underpowered, unprintable, unpublishable. I think that contract will be easy to solve. We then get into the whole issue of data integration and data mining, and that's where Tel Aviv University will come in, as well as others. We get into causal modeling, because we'll no longer be talking about correlative statistics, we'll be talking about causal, rule-based classifications. Then we get, therefore, we generate biological signatures of disease, and with that we go to see our geneticist and omics colleagues and we say, this is the vector in this patient, or in this group of homogeneous patients. What system, what biochemical or physiological system is affected in this disorder fundamentally? We'll go to see our clinicians who will now be doing deep phenotyping and asking two sets of questions. One is, what is the variability of expression in a group of biologically homogeneous people? And are there easy ways of detecting differences between them without having to go to these high technology aspects? And of course, doctors will continue to perform their fundamental function, which is to interact with the patient, to give them the faith, to give them the trust, reciprocally, in order to be able to help them at the human level. So we'll go to the pharmacologist then and say, well, this is the system the omicist has found. What drugs do you already have in your pharmacopoeia? And I tell you, they have all the drugs that are necessary to give a cocktail to modulate the function in the system. I've recently seen an experiment where a drug was given at dose 1 and had an effect. Another drug working on the same system was given at dose 1 and had an effect. And then both were given at dose 0.1 and the effect was 100 times higher. This is just an explanation of what we all know, that this is a complex system and not a linear system. Adding 1 and 1 can mean 100. Adding 1 and 1 can mean 1. Adding 0.1 and 0.1 can mean 100 in this sort of context. And then finally, we'll go to the clinical trials where we will have groups of patients that have already been defined biologically. So we will not have these errors creeping in. So this is the MOE. You all know what an MOE is? No? The mother of all experiments. So in the mother of all experiments, we managed, now just to show you feasibility, I've got to show you feasibility here. I went to 3 French groups and got 5,566 healthy controls of the right age and 912 Alzheimer patients. Now that in itself is a miracle. Everyone knows about the French and their data. It's mine. They've been collecting this for 10 years and have got a few publications. They gave it. They were convinced. So we've got data on various types of scans. We've got clinical scales and measurements, We've got between 500 and 500,000 and a million SNPs. We have various proteins, including APP, tau, presenilin, and so on, from the CSF and from the blood. And the data are incomplete. The data are not normalized. Some of the data are in terms of labels rather than numerical. A lot of the data are numerical. A lot of the data are highly complex. A lot of the data have enormous weight. And yet there are industries and sciences, CERN, the nuclear industry, the aeronautical industry, the chemical faculties, the physics faculties, that deal with these problems every day, and we as life scientists and we as medics haven't got the faintest idea. We're so far behind, we're Neanderthals. Typical Neanderthals who are trying to do things in the old way when everyone else is advancing extremely rapidly using the tools that have been produced by computer science. Now, one of the hopes that we have, of course, is that the techniques that have been used here and are championed by Myra and her colleagues are going to be helpful, and we're looking at a number of these techniques. Some of them are in the commercial domain already, some of them are in the academic domain. Here's one which is called HyperQ, and essentially it collects these types of data, it pre-massages them, it puts them through specialized separate dedicated calculation networks, which aren't supercomputers. So imagine transferring this to supercomputers. That's another little contract, which is a win-win situation. You've got a nice tool here. It takes a long time to do. Wouldn't you like to do it on a supercomputer? We've got a supercomputer. Yes, please. Boom. In they are. And of course, we can test lots of them in this way. And they give us back rules. Now, what's a rule? A rule is a parameter, or it's a feature which is parameterized. So it could be between the age of 60 and 65. So we have found groups of people who are between the age of 60 and 65 that have got amyloid precursor protein between such and such a concentration, that have this and this feature clinically, that have this and this area which are atrophied to this and this extent, and they form a homogeneous group. And they have to have all of those rules. And you'd think, how many of there are there? How many of these groups are there in, say, 200 subjects? Think about it. We'll get to the answer in a moment. Henry showed you this. This is where we interact with the Human Brain Project. Because what was shown here was that there are data sources about various features where you get the data and you develop the principles. What we're going to be doing is the same thing but in diseased patients. And so we will be introducing here the diseased data and the diseased principles. And then what Henry will do on his simulation is plug these in place of the normal and see what the model predicts. And what the model predicts will then be contrasted with what the clinician finds and with what is found in the accompanying tests. So this will form a form of validation which will be extremely useful. If there's not congruence, then we're into the iterative phase. We go back, we find where there's missing data, and we continue the process. And that's why this process is likely to be a cultural and intellectual process which will take many years. But just to try and convince you that getting mechanistic and causative diagnoses in the form of disease signatures will be helpful, here are the preliminary results from 200 subjects taken from the ADNI database. And what you see is that 199 of the 200 were classified in 6 groups. 6 groups which had between 5 and 9 rules describing them, where 199 of the people fell into one of the 6 groups. In other words, had the full set of rules relevant to the particular group. Now, we've tried to do this on 500 subjects. We're iterating continuously. That's why this is taking a little time. You can see here rules for Alzheimer's and rules for normals. This looks as though it's going to be Alzheimer's disease because it's got APP and ApoE4 associated with it. You can see that the normals have certain SNPs, that's the genetic ones, the black ones. The different types of Alzheimer's have different types of SNPs associated with them. The CSF and proteomics are in pink and purple, and you can see the orange is the PET data, showing that the different dementia types appear to have atrophies in different distributions, something I've already shown you in the group studies. So it's looking very much as though we're on the right track. We're going to continue on this track, and in the end, of course, we'll be left with philosophy. And this is my contribution to philosophy, care of the New Yorker, and I hope very much that you've enjoyed following my logical, linear train of thought. But the thought I want to leave you with at the end is it's not going to be linear at all, and it's going to involve lots and lots of people interacting and producing every bit as much intellectual power as they have in the classical reductionist technique. But now it's going to be predictive, it's going to be simulation, and that's what we need for organic matter, and that's what we're going to need for understanding our humanity through our brains and our diseases through our brains. Thank you very much.
So thank you, Richard, and we are very short in time, so just, let's say, 2 questions. Either? איפה?
You've mentioned therapy. I wonder if you are talking about occupational therapy, physical therapy, and in that sense, I would like to address that actually medicine is one thing, but unless you are doing a lot of activity with the person, and I have unfortunately an experience with my mother, and I came back here in order to take care of her. So music is the fantastic thing to start with, familiar songs.
Yes, I lost my mother in December after 5 years of the same disease. So I fully understand what you're saying and why you would focus on a therapy that clearly had a lot of impact on your own mother's well-being. I think one of the great problems we have as medics, and particularly in our interaction with scientists, is that the individual is the primary source of our concern. And yet, we have to have science, because without the science, we don't have the knowledge on which we can base the treatment of the individual. So I could very happily conceive of dementias which perhaps primarily started in the auditory and associated auditory cortices, and which could be entrained in some way, and where compensatory mechanisms could be strengthened with music. I could even entertain that sometimes it could be Iron Maiden, and at other times it could be Mozart. But I don't believe that should be a generalization across all dementias, because I think that in certain cases there will be dementias which we may be able to treat in very physical ways. There may be dementias in which different systems need to be treated with different strength drugs or other rehabilitative techniques. So I take your point, but I think we're going to need to look wider.
I think we'll cut it shorter now because we are really late, and I want to thank everybody who stayed with us till the end without a break. I'm sorry, and I hope for more like that. Thank you, Henry. Thank you, Richard. Thanks, everybody.